Hi, in this video we will look at how to set up a paper chromatography experiment and how to calculate the RF values for the components in a mixture. If you need more help or would simply like some practice questions, then please click the links in the description below. Chromatography is a technique which can be used to separate out mixtures of substances, and it can also be used to identify the substances present in these mixtures too. Now, there are many different types of chromatography, but in this video we will be looking only at paper chromatography. Paints, dyes and inks usually consist of a mixture of many substances, and paper chromatography is an excellent method to use to separate out the substances in these mixtures. For example, imagine we had a green dye which we wish to investigate and find out how many different substances are present in it. Well, to carry out a paper chromatography experiment to answer this question, the following method could be used. Firstly, a pencil line is drawn on a piece of chromatography paper about 2 centimeters from the bottom. This pencil line is often called the baseline. Next, the substance to be tested, in this case the green dye, is spotted or pressed onto the paper a few times using a capillary tube to increase the concentration and the amount of substance present. Next, the chromatography paper is suspended in a beaker or a tank containing a suitable solvent. That is one which will at least dissolve or partly dissolve the substances present in the green dye. If the solvent doesn't dissolve the green dye, then none of the substances in it will move off the baseline. Now, it is very important when setting up a paper chromatography experiment that the solvent is below the baseline. Once the chromatography paper is in the solvent will soak into the paper and start to travel towards the top. As the solvent passes over the green dot, the substances in it will dissolve and start to rise up the paper. We will explain later in this video how chromatography is able to separate out the different substances present in the green dye. Once the solvent has almost reached the top, the paper should be removed from the tank or the beaker and left to dry. The dried paper with the dots is often called a chromatogram. From looking at the chromatogram, you can see that the green dye consists of two separate substances, a yellow dye and a blue dye. But how are they separated on the chromatography paper, and how does chromatography work? Well, to answer that question, we need to introduce two new terms. These are the mobile phase and the stationary phase. Now, a phase is just another word for a substance that is either in a solid, liquid or gas state. The mobile phase in paper chromatography is a solvent, which is usually water or ethanol, while the stationary phase is the chromatography paper. As an example, to explain how chromatography works, consider what happens when the solvent soaks into the chromatography paper in the image shown. Now, as the solvent soaks into the chromatography paper and passes over the red dot, the dye molecules present in the dot can do one of two things. They can either dissolve in the solvent, that is the mobile phase, and travel up the chromatography paper towards the top, or they can stay attracted to the paper molecules, that is, stay in the stationary phase. It is important to realise that as the solvent rises up the chromatography paper, the different dye molecules present will be continually moving from the solvent to the paper and back again. That is, from the mobile phase to the stationary phase and back again. This means that the more time the dye molecules spend in the mobile phase, the further they will rise up the paper, and the more time the molecules spend in the stationary phase means they will not travel up the paper as far. So basically this means that the pattern of dots produced in a chromatogram depends on how much time each of the component dye molecules spend in the mobile phase and the stationary phase. In order to try and find out which food colorings were present in a food sample, the chromatogram shown on screen was produced. Here, spots of green, red, blue and yellow food colouring were placed on a piece of chromatography paper alongside the food sample to be analysed. You should see that 3 of the food colourings, the green, red and blue, were pure substances. That is, they contained only one substance, since only one dot was produced on the chromatogram for each of these 3 substances. But the yellow food colouring is a mixture of different coloured substances, a red colouring and a green colouring, which were produced as 2 dots on the chromatogram. Now the food sample contains 3 substances, since 3 dots were produced. The blue and red dots in the food sample line up with the blue and red dots for the food colourings, so this likely means that the food sample contains both the red and blue food colourings. But there is also one unidentified, unknown substance present in the food sample. Now, to compare how each component moves in a chromatogram, we use a measure called the retention factor, or RF value. This is calculated using the formula shown on screen, with all the distances involved simply being measured using a ruler. Now the distances moved in centimetres by the red, green and blue food colouring are shown. These distances, as you can see, are all measured from the baseline, and the distance moved by the solvent is often called the solvent front. In this particular chromatogram, it is 10.5 centimetres. So to calculate the RF values, simply put these numbers into the formula as shown, and this will give you the RF values for each of the food colourings. You can see that the larger the RF value, the further the dot moves up the chromatography paper. Now the unknown substance, that is the orange dot in the food sample, we could identify it by measuring its RF value and look it up in a database to confirm its identity. However, care is needed with RF values, as other substances in a database could have these values, so it would be useful and wise to repeat this experiment using different solvents and then look up the new RF values produced against a database using that particular solvent. Hi, thanks for watching. If you found this video helpful or would simply like more information, then why not visit the website that accompanies these videos? The links are in the description below. If you enjoyed the video, then please consider liking and subscribing. Thank you.